Scientists have devised a virtual reality platform for lab animals. Let that sink in. Zebrafish have swum with the aliens from the video game Space Invaders, mice were afraid of virtual heights, and fruit flies circled illusory obstacles. This new holodeck for animals can help researchers see how freely moving animals respond to a variety of illusions, work that could help scientists better understand human genes and brain circuitry, researchers say. The researchers, from the Vienna Biocenter in Austria, detail their findings in today's edition of the journal Nature Methods. Image Straw Lab researchers controlled a fly's position, red dot, and flight direction by providing visual stimuli. Freemover immersed animals in arenas, where the walls or floors were computer displays. Each screen depicted photorealistic images that accounted for each animal's perspective as it walked, flew, or swam. Up to 10 high-speed cameras monitored the precise 3D position of each animal. Freemover then updated its video imagery within milliseconds of each animal's movements to create the 3D illusion that they were moving in environments that changed in response to their actions. The researchers compared Freemover to the holodeck, a fictional environment in the TV show Star Trek in which humans enter a computer-controlled virtual world, says Andrew Straw, a neurobiologist at the Vienna Biocenter who is co-senior author of a paper detailing the study. They can freely move, have no need to wear special clothing or headgear, and are immersed in a computer-controlled environment, which can be made completely realistic or arbitrarily unrealistic. Image and Graphics Department. The researchers tested Freemover on mice, fruit flies, and zebrafish, 3 species commonly used in lab research. The virtual landscape with which these animals interacted included vertical pillars, floating rings, checkerboard floors, virtual plants, and a swarm of digital aliens from Space Invaders. They even had distinctive portals that could instantly alter the virtual environments to make it seem as if zebrafish swimming into them had teleported elsewhere. The animals apparently found the illusions realistic. For instance, fruit flies circled virtual pillars just as they did real ones placed in the platforms. Moreover, mice generally avoided tracks that looked as if they were suspended at great heights, just as they would in real life. Image Straw Lab. The animals also changed their behavior in response to illusory animals. For example, zebrafish normally circle the periphery of their fishbowl near the screens, but when teleported into settings with swarms of Space Invaders, the zebrafish tended to move toward the middle of the fishbowl. We wanted to study collective behavior because that is something incredibly difficult to do with real animals or with robots, Strauss says. We wanted to show how real fish respond to the motion of a swarm of simulated agents, and to show that we could create a hybrid biological computational swarm. In addition, the researchers developed a photorealistic model of a swimming fish and showed that real zebrafish most reliably followed the digital fish when the virtual animal matched its swim direction to the real fish. The fact that researchers can vary the appearance of virtual animals from cartoonish to realistic will allow experiments to test how important the exact visual appearance of other animals is as opposed to, say, the pattern of motion, Strauss says. This new platform will let scientists investigate animals as they behave relatively naturally and unrestrained by conventional VR gear in realistic virtual environments they can manipulate extensively. By tinkering with animal DNA or brains in such experiments, the researchers can learn what role certain genes or brain circuits play in these animals, and potentially in humans as well. Brains evolved in the real world, and to understand how and why neural circuits process information in the way they do, we need to understand them in this context, Strauss says. Straw notes that humans would notice several imperfections with Freemover. Primary amongst those is that our system does not create 2 distinct views for the 2 eyes, and thus the stereo cues important for depth perception would be gone, he says. However, Straw notes this is not a major concern with the animals they are experimenting with. The eyes of these animals are so close together that the differences between the view from each eye are limited. Straw's lab is now conducting experiments where they can silence the activity of single brain cells in fruit flies and examining the roles these cells play in the insect's behavior in virtual reality.